लाइव
योगी
Immense pleasure and honor, I welcome you all to this national conference on advances in personalized medicine, tailoring pharmaceutical inventions for optimum health. Of innovation and healthcare, we witness a paradigm shift from one size fits all approach to a more patient-centric, individualized form of treatment. Personalized medicine, driven by advances in genomics, data analytics, biotechnology, have paved the way for more effective and targeted pharmaceutical interventions. Today, we are not just treating diseases, we are treating individuals based on genetic makeup, lifestyle, and environmental factors, leading to enhanced therapeutic outcomes and reduced adverse effect. The objective of this seminar is to explore the latest breakthrough in personalized medicine, to understand the role of emerging technology such as artificial intelligence, big data, CRISPR, gene editing in refining treatment strategies, to foster collaboration among researchers, industry leaders, faculty and students to accelerate the translation of research into clinical practice, ensuring that future professionals also benefit from these advancements. The impact of personalized medicine is profound. We are now able to predict disease, design targeted therapy, and optimize drug dose, ensuring that every patient receives the right treatment at right time. With this approach, we are moving closer to the vision of precision healthcare, an era where medicine is as unique as individuals receiving it. I would like to extend my heartfelt gratitude to our esteemed speakers whose contribution to this field have been instrumental in shaping its growth. I also thank the organizing committee, our sponsors, and all the participants who have come together to make this event a reality. Your dedication and commitment to advancing healthcare inspires us all. As we embark on this enlightening journey today, let us engage in meaningful discussions, challenge existing paradigms, and envision a future where personalized medicine becomes the standard of care. May this conference serve as a catalyst for new ideas, collaborations, and innovation that will ultimately benefit patients worldwide. Once again, I welcome and I wish you all productive, insightful conference. Thank you all.
Thank you, respected ma'am. I now invite our respected Vice Chancellor, Professor Dr. K.L. Verma sir, to share his words of wisdom. Let's welcome respected sir with a round of applause.
बहुत ही प्रसन्नता हो रही कि हमारा विश्वविद्यालय 2 दिवसीय नेशनल कॉन्फ्रेंस करवा रहा है जो कि एडवांसेस इन पर्सनलाइज्ड मेडिसिन ट्रेनिंग फार्मास्यूटिकल इन्वेंशंस फॉर ऑप्टिमल हेल्थ पर आधारित है। इस उद्घाटन समारोह में और प्रथम सत्र में हमारे बीच उपस्थित हैं गेस्ट स्पीकर के रूप में डॉक्टर श्रीमती निर्मल मिलिंद कासेकर जी। बड़ी खुशी हो रही है कि आप हमारे बीच हमारे स्टूडेंट्स को, हमारे टीचर्स को इस संबंध में विशिष्ट व्याख्यान के माध्यम से जानकारी देने उपस्थित हुई। हमारे बीच उपस्थित हैं मिस्टर राजन हलमकर जी, विवेकानंद कॉलेज ऑफ फार्मेसी के। हमारे विश्वविद्यालय के रजिस्ट्रार डॉक्टर आर शर्मा, डीन डॉक्टर मोहसिन हसन, विनीता प्रोफेसर कीर्ति शर्मा, प्रिंसिपल ऑफ द फार्मेसी डिपार्टमेंट, प्रोफेसर वनिता लोखंडे, हमारे सभी शिक्षक साथियों एवं प्रिय विद्यार्थियों, बहुत ही विशिष्ट विषय लेकर 2 दिन का कार्यक्रम आयोजित हो रहा है और इस पर अभी सोइनियर का विमोचन हुआ है, अब्स्ट्रैक्ट है पेपर से। अच्छा लगता है कि जब ऐसे डॉक्यूमेंटेशन के माध्यम से हम अपने रिसर्च को, हमारे विश्वविद्यालय द्वारा किए गए इस कार्य को हम डॉक्यूमेंट करते हैं। मैं तो इस विषय का विशेषज्ञ नहीं हूँ, लेकिन कुछ जो बातें इस संबंध में हो सकती है एक लेमन की तरह, एक सामान्य जनता की तरह वो करना चाहता हूँ। दवाइयाँ बहुत बनी, प्राचीन काल से हमारा देश इस मामले में बहुत आगे रहा, समय परिवर्तित होता गया। एलोपैथी हो, चाहे वो आयुर्वेद हो, चाहे होम्योपैथी हो, इसमें बहुत कुछ प्रगति हुई और लगातार इस प्रगति के दौर पे देखा गया कि नई नई समस्याएं आई, उसका निदान हमारे विशेषज्ञों ने किया। समय के साथ यह परिवर्तन आता गया। बात इस बात पर होती है कि जेनेटिक्स क्या है, जेनेटिक्स में क्या एडवांसेस हो रहे हैं, लाइफ स्टाइल बदल रही है, हर व्यक्ति का खान पान, रहन सहन और जिस क्षेत्र में वो रहता है उसके वातावरण के अनुकूल की परिस्थितियाँ और उसका व्यवहार, उनका सब कुछ बदलता जाता है। और मैं मान के चलता हूँ कि दुनिया कहती सबसे कठिन काम है मनुष्य को, उसके शरीर को समझना। आज भी हम कहते हैं कुछ चीज़ें बना सकते हैं, लेकिन कुछ चीज़ें जो कि मनुष्य से ही या जीव से ही प्राप्त होती है, हम नहीं बना पाते। आज भी समझना जो कुछ हुआ है वह बहुत कम हुआ है, बहुत कुछ जानना। विश्वविद्यालय का काम होता है कि हम अपने विद्यार्थियों के लिए, हमारे शिक्षकों के लिए ऐसे आयोजन करें जिसमें विशेषज्ञ आए, वो अपनी बातें रखें, नए शोध के जो आयाम खुल रहे हैं उसके बारे में जानकारी दें और हम बच्चों को मोटिवेट करें कि भविष्य किस तरफ जा रहा है और आपको क्या करना है। लगातार हमने बहुत सारे अलग विषयों में कॉन्फ्रेंस कराए, लेकिन फार्मास्यूटिकल्स का जो महत्व है, जो फार्मेसी डिपार्टमेंट को मैं बहुत महत्व देता हूँ। मैं तो जिस विश्वविद्यालय में पहले था, पंडित रविशंकर शुक्ल विश्वविद्यालय रायपुर का वाइस चांसलर रहा 5 साल, मैं संयोगवश बताता हूँ क्योंकि इस विषय से जोड़ रहा है। मैं शान से कहता था कि सभी डिपार्टमेंट जो है सबसे टॉप में फार्मेसी डिपार्टमेंट है। हमारे विद्यार्थी भी इतने आगे बढ़े कि आज फार्मेसी काउंसिल का जो एकेडमिक हेड जो है धीरेन्द्र सिंह मेरा स्टूडेंट था। ऐसे हमारे बच्चे करेंगे, मैं इसलिए कह रहा हूँ। नाइपर के जो डायरेक्टर है मेरा छोटा भाई समझिए, मैंने ही उनको रिकमेंड किया और रिलीव किया और वो डायरेक्टर हैं, उसी यूनिवर्सिटी के प्रोफेसर हैं जो फार्मेसी काउंसिल के पहले वाइस चेयरमैन भी रहे हैं। स्वर्णलता सराफ भी एग्जीक्यूटिव काउंसिल में फार्मेसी काउंसिल का तमाम उसी रविशंकर यूनिवर्सिटी की बात कर रहा हूँ, जहाँ कम इसलिए कि वैसा वातावरण निर्मित किया। और मैंने कहा कि नंबर 1 कहता था सभी डिपार्टमेंट से ऊपर में, और उसकी
but they have almost similar, they overlap each other, right? So, patient's unique profile we identify, we then there is a reduction in treatment failure and we get good results because we minimize side effect. We minimize side effect. Now, all my students, when I deliver some session to them or my audience, I always tell them, you keep with you pen and paper, but nowadays आप मोबाइल का नोट भी खोल सकते हो. Wherever you find some interesting point, you can note down. Our memory is very very volatile. Only my request is keep all mobiles on silent mode, okay? And don't use any other Google or something. Just concentrate because you are here. I am also here. I was sitting with this presentation for last yesterday 1 o'clock because I want to deliver something good to you, right? All my dear students, yeah, friends, rather, you're with me. Let's see. Let's see. So what is the difference here? It's a popular basic medicine. एक ही मेथड सबको. ऐसा हो पेट दर्द हुआ ये गोली खा ले बात खत्म. But here we are trying to find out differences and prepare the medicine. Hello sir. Hello sir. No problem. So and then we are formulating the medicine according to the need of an individual. Your needs are different, my needs are different. Rightly quoted by PC sir that एक कैंसर का ट्रीटमेंट है, लाइक केमोथेरेपी है, रेडियोथेरेपी है, सब मेडिसिन है, सबको क्यों नहीं सूट हो रहा है वही डोज वही रेजिम? यस और नो? करेक्ट? क्यों नहीं सूट हो रहा? बिकॉज वी आर डिफरेंट, वी आर यूनिक. I would rather say we are unique, right? So that uniqueness we will find out and then we will prepare medicines for that unique disease. So there are elements of personalized medicine. I will touch 3 elements and there are different sub-elements of personalized medicine. If I want to go ahead, see, here are the students who are sitting. I think they are B.Pharm students and what I got to know that your college had applied for M.Pharm courses. So shortly your college will start M.Pharm, then they will get admission into masters, say pharmacology, pharmacochemistry, yes or no? Not this college, maybe other college, right? Right or not? Fine. So now in that case, you should know exactly what to select, what are your likings, and your liking will depend upon your understanding to that particular point. And nowadays, irony is, my dear students are so busy and they are just busy with reading their syllabus, their question answers, answering and putting them on answer papers and forgetting next day. Yes or no? Am I correct? Yeah. So that will not help, my dear students. Whatever I will tell you technically, there are many stalwarts who will be discussing today and tomorrow on this particular. I will just touch and go every small topic. I am here basically to give you the gist and to motivate you all, my dear students. Now, so the first element of understanding individual is genomic. What is genomic? May I ask you all? You are made up of different DNA. पता है? कितने होते हैं DNA? Billions. Okay. एक DNA का लेंथ कितना होता है? Don't Google. यहाँ पे देखो straight. Okay. हमारे बॉडी के अगर पूरे DNA साथ में ले लिया और एक लंबा चौड़ा लाइन बना दिया, कितना बड़ा होता है? एक मिनट, क्यूरियोसिटी रेस कर दिया है मैंने. आंसर एक चीज का बता देती हूँ, दैट इज 2.1 बिलियन किलोमीटर. फर्स्ट का आंसर आप फाइंड आउट करिए. पूरा सब जोड़ के अगर हमने ले लिया तो वो गॉड इज सो ग्रेट, ही हैड क्रिएटेड सच अ ब्यूटीफुल क्रिएचर. उसके बाद में उसने कुछ क्रिएचर बनाया नहीं है शायद से, एंड वी आर सो कॉम्प्लिकेटेड. गॉड मेड अस डिकेड बैक, राइट? एंड वी स्टार्टेड अंडरस्टैंडिंग आवरसेल्फ नाउ. So हम कितने backward है human being and still we think we are so great. Anyways, so genomics is study of genomics is very important. Now when you have data, उसको analyze करते तो आना चाहिए. One very good friend of mine, he is an senior accountant of one firm. तो उन्होंने लिया PhD में admission. See, my talk will be with story. So my friend, he took admission to PhD. And then he collected the data. The topic was something like higher education and then hierarchies and then appointments of people in higher education state, administrative position. He collected data, he collected data, and then he went to the guide. And he collected data from very reputed institutes, national importance institute, n number of. So one fine day he sat in front of his guide with his, I think Bangalore में उनका पूरा data लेके गए, and they were like, he was reading, he was looking at him, he was reading, looking at him, he was reading, looking at him. He finished his whole data collected, and then he politely asked, Ramesh, very good data. यह research भाग है. My dear students, we mistake research as data collection. उसको include करना जरूरी होता है, उसको analyze करना जरूरी होता है. तो big data of the genomics will not help you. उसको analyze करना है, उससे सब conclusion पे आना है. There are 5,000 DNA in your body of billions of DNA who are responsible to cause disease. In 1998, Dr. Mrs. Shubha Phadke was first medical graduate. She went to Lucknow, I think, yeah. If I am wrong, I don't know which place. But then she took admission under Mr. Dr. S.S. Sir to study genomics. 1998, my dear students, जब किसी को पता भी नहीं था genomics क्या है, DNA क्या है, reporting क्या है, हंसे रहेंगे ना उनके ऊपर. I'll tell you my story. When in 1994 I took admission for pharmacy at Bharati Vidyapeeth, one fine day, one uncle I met when I was just walking down to the college. He was like, "Oh, MR Pandachi मुलगी pharmacy ला admission घेतलं." म्हटलं हो सर. मग compounder होणार का? That was the scene long back. You all are so fortunate. अभी वैसे नहीं है. People respect you for your knowledge, not because you are son of somebody, a daughter of someone. Yes or no? Because you are a student of some college. Okay? So kindly attention, my dear students, and see to that you will reach to certain end. Not just you will not stop your education till D Pharm or B Pharm, masters, you do PhDs. The next speaker, my beloved teacher, my guide, my mentor, she will be definitely guiding you about AI in this. I have few slides, I will skip those because anyways madam will be dealing with that particular topic. She is stalwart when it comes to process form development. So what? Takeaway now? Data, analyze, conclude something. And then everybody talks about proteomics, genomics. So this omics. Now, more comprehensive understanding. And last is, you then go for your, you find out the target and you go for dosage form development, you see the effects and side effects. Step 2, now we'll move ahead. So, now third element is this advanced diagnostic and technology. Now here, what I apply here, you can see early detection very important. In cancer, everybody know, yes or no? So what early detection means here? What early? Cancer में कितने जल्दी early detection हो सकता है? या फिर any disease है, उसका early detection कहाँ पे हो सकता है? Prenatal stage. Prenatal stage, there are a lot of research which are available. I just told you 5,000 DNA, which are disease-causing DNA, can be evaluated at just rupees 20,000. But the laboratories. Registered certified laboratories, wherein you can find out what your body is prone to. आगे आपको क्या हो सकता है? There is chances of. That is what happening here in the world of research. Okay. Then targeted therapies. Simply, if you have a headache, then why to take orally? Can you give it locally? So here advanced targeted about AI machine learning everywhere now there is AI. But chat GPT तो आपको पता ही है, very well known. ऐसा ना? Correct or not? My daughter, she uses chat GPT. So I prepared the slides and then I said I wanted to see कि chat GPT करता क्या है. Many of my students are also like chat GPT बहुत easy है. दिखाओ ये टॉपिक डाल के क्या इंफॉर्मेशन दे रहा है. चैट जीपीटी gave I think 9 slides. But I was like, ये 9 slides में आपको क्या समझ में आया? What is your understanding? They will give you bullet points. But अगर आपको स्टडी करना है, सोचना है कि आगे मुझे क्या करना है या फिर if I can do something, आपको in depth स्टडी जरूरी है. In depth. Then biobanking, storage of this data, very very important. Let's go to the step 3. See, when you are into any process, it is a stepwise process. जैसे for example, when you are born, you go to your kindergartens, then you go primary, secondary, and then you go to higher levels, right? Higher education, right or not? So everywhere there is a stepwise study. So now step 3 say data privacy, very important, and it's a very big problem. See, if I am giving my genomes for study, 5,000 DNA been evaluated, and then I know because the reports are with me, but then I don't want others to know, na? Are you getting my point? What I am not to is my personal information, and maybe only should be known to my doctor. Why others should? And if somebody breaks into that system, data privacy becomes very, very dangerous. Informed consent, like for formulating any dosage form, हमें preclinical clinical studies करना है. Yes or no? Final year students, yeah, जाग रहे हैं. I am very loud, so you should be also loud. नाश्ता किया है? अरे नहीं किया है तो कर लो बाद में. अभी तो बोलो, जाग रहे हो? Yes. Thank you so much. Okay. So I will just keep in front of you one case law. Fortunately, I am a law graduate of Mumbai University. So on this case law, I can take one whole session, 3 hours. Okay? So like medical, so here, informed consent, why it is important when you are giving medicine to the people, patient, they are not known to the effects and side effects, they are dying. It is good or bad? No, yeah, it is very, very bad, right or not? So this was done by Pfizer in 1996. For an anti-meningeal drug, Provan. A lot of fine was been, they paid to the Nigerian people. So, I'll take you to the different milestones of this personalized medicines. Can you read this first? Discovery of DNA structure, when has happened? Can you, anybody tell me who discovered DNA? Yes, sir.
Marcellin Greek.
Thank you so much. A big round of applause. Thank you so much. So now, 1953, where are we now? Where are we now? So still, many things are to be done. Many things are to be done, and who will do it? People sitting across, you people, researchers, future of pharma. Okay? Then birth of, there is a pharmacogenomics came into existence in 59. In this particular span, human genomic projects were undertaken. Then this FDA approved this list herceptin. You know what herceptin is? Which drug it is? Pharmacy, final year students? At least? Okay. It is a drug which is used to treat breast cancer. Only to the HER2 positive patient this drug is effective. HER2 negative people cannot. Okay? Yeah, clinical pharmacologist. I am correct, ma'am? Yeah, correct. Then there was an introduction of pharmacogenomic testing. FDA launches precision medicine initiative. Now, you know CDSCO? Final year at least diploma students, final year BPharm students. What is full form of CDSCO? Yeah, correct. So what they do? They find out whether drugs are safe to us. Yes or no? They do inspections. Correct or not? A lot of data has been asked from the people who are into the research. Then thank you. Then there is a rise of T cell therapy. Now, madam will definitely discuss it because she will be touching to the AI. Okay? And then AI and big data. Depersonalizing the personalized medicine. Because you need data. You need a lot of data. So, actually I prepared it, but I think ma'am will be taking care of it, so I will just do the run away a little slides. One more. Yes. So, first is drug target identification. Second will be molecular design. Third will be drug repurposing. Available drug repurposing. Very cost effective method. Clinical trial optimization. Because it is there, trials are there, but now if you go for this DNA and all, you need to optimize this process. Drug safety and side effect predictions. What you do in this clinical studies? You predict, right or not? Right? So ultimately, big data and your AI, what they do? Analyze the, what they do next? Identify the pattern, how your DNA is, how it will react, and predict the treatment. So this is takeaway. Okay? So I think this is AI based. AlphaFold, which is developed by DeepMind, talk about all AI design. Now this particular system I developed, been used to predict the protein structure of which virus? COVID virus. Okay? Why we got COVID vaccine so early? Because they had used this particular system. I think Madam will talk about this, but still, I would say this CAR-T cell therapy for cancer. You know, my dear students, when we treat cancer, now, I know many people who treated, cured, because I'm associated with MGM, and also we have many relatives or friends, right? So we know the things. So what they are, either as Sir said, ड्रग इफेक्ट हो रहा है या नहीं हो रहा है, और इतनी बार उनको मेडिसिन लेने जाना पड़ता है, कीमो लेना है, रेडियोथेरेपी लेना है, ये सब 15-15, 20-20 कीमो लेना है, 30-30 रेडियोथेरेपी लेना है, ऑपरेशन तो हो गया है वो अलग बात है, एक ऑर्गन लूज कर दिया है वो अलग बात है. What about this? I have highlighted something. Next slide. Is this single infusion only takes 30 to 90 seconds, minutes. Indeed, it was time saving, but then what was the drawback? You read first line? Cost. High cost. So like where we stand, my dear students, basically pharmacology, pharmaceutics, all branches, where we stand and what we should look into is how we make cost effective. So basically breakthrough अगर हमें देखा तो real time health management system this personalized medicine has given to us and predictive. You can predict. होता है ना आपका क्या बोलते हैं? they tell you कल बारिश होने वाली है. कौन बता दे? हाँ, but कल बारिश होती नहीं. जिस दिन बोलेंगे इन मुंबई में आपका जिस दिन बोलेंगे बारिश नहीं होगी उस दिन बारिश जरूर होगी. जब बोलेंगे बारिश है और हम लोग घर में है तो बारिश. Jokes apart, but then predict. When you know what is ahead, prevention is better than always. So, what are the future prospects, my dear students? Personalized treatment. Okay. Next, where you will be developing a drug. Third, what can you read? Yes. Next is yes. Then next, enhance research. And here, why I haven't touched all this slide because this data is there. Enhance research. So, at my time, the doing research was still okay. We could do it because we had data. There was computers. Many years back, when they were not coming to research, you are a doctor. I don't know when you did your PhD. Data gathering, compiling was a big task. Now we have big data. We have all things available with us. We have AI, we have computers, we have everything. So can we do good research? Yes or no? Yeah. And improve patient engagement, because ultimate aim of every research guideline is we want to safeguard our patients. Yes or no? Best treatment available. The way sir said, we are giving best students. Right? We are giving best to students, and then these students will be best. And they will serve nation. Yes or no? Correct. And finally, at reduced cost, which is not now available because it is very costly. But my dear students, if you start, if you think, then definitely you can. But then still challenges again. Okay, so what are the challenges? Data privacy and like, now this, can you predict something? Abhi discuss करेंगे. Yes, please. Affordability, accuracy and affordability. Number one is affordable. You can create any number of things, but affordable नहीं, कौन खरीदेगा? खरीदार चाहिए कि नहीं चाहिए? Yes or no? And what, my dear students, a very very important collaborative work. See, there are differences. Medical field का education अलग है, pharma अलग है. Then if you talk about policy makers, their profession is different, their expertise is different. But if you want to come out with a good result, everybody should work collaboratively. Take an example. You have a system where you have heads, like you have HODs, you have a principal, you have, and then VC, pro-VC, and so many people. They make policy, they percolate it to you, your teacher implement, and you go ahead. Yes or no? Simple. But then this particular collaborative work and expertise should work really with the one thinking, with one goal. If you sit and say, I am the best, chalega, I will not give you my knowledge to anybody. It happened with Ayurveda in India. Why Ayurvedic knowledge is now not available? There were many remedies which were available in ancient India, but why not known to us? Because many Vaidus, they died, but they didn't percolate the knowledge to the next generation. चलो सिंपल बताती हूँ, थोड़ा ज़्यादा हो गया. आपने कभी डैडी को सुना है बोलते हुए, माझ्या आईसारखं जेवण तुला नाही बनवता येत? सुना है? Yes. Because your mother might have learned many things from your grandmother, meaning daddy की मम्मा, सासू माँ. But सासू माँ थोड़े से ट्रिक नहीं बताएंगे, उससे वो खाना उतना टेस्टी नहीं बनता. सिंपल है. जब आप तड़का देते हो तो उसके ऊपर ढक्कन रखना है. And people like we will keep open and दूसरा डाल डालेंगे, सब डालेंगे, तड़के का स्मेल चला जाएगा, वो टेस्ट नहीं आएगा. Correct or not? Yes or no? So percolate knowledge. Why I am standing here in front of you, though I am a principal, don't you feel I have the responsibilities there? But when I see you all sitting, fresh faces, I find you all are like chargers. Now I am getting energy. You don't understand? I am here to get energy. And I am really getting good energy because at least one person gave answer. I didn't ask questions also, but you are so connected with me. I can see nobody is looking into mobile, nobody is using mobile. So thank you so much again for that. So conclude. See, देखो अभी हो गया खत्म. थोड़ा से 2-3 slide. Are you with me again? Yes. So to conclude, I will just talk about this, or I will just read simply these 5 sentences, very self-explanatory. Personalized medicine is transforming what? Healthcare. It enables improved patient outcome. Clear है? कुछ expressions required? No. Continued innovation and collaborations are right. They're important. Empowering patients with personalized insights of product health management is key. And patient know मैं क्या ले रहा हूँ. मेरे लिए I'm important. मेरे लिए एक दवाई बन रहा है. Yes or no? Yes. क्या? सबको पसंद है importance लेना. Yes or no? कोई आपको importance देगा तो आपको पसंद नहीं. Yes. This shifts from one side spread to all to the personalized, which is happening now. But so what prediction in 22-24 the market is 5.3 1.7 billion in 2030, it might reach to 869.9. But who will do that? Any commerce graduate can do that. Arts graduate can do that. Simple science graduate can do that. They can, if they have specialization. But pharma students, can you? So, this slide says, what is this slide? Just a slide to motivate you all. Research is what? To see what everybody has seen. देखा तो है सबने, but उसको जाना है, अप्लाई किया differently. You देखा आपने वो मूवी है अक्षय कुमार और विद्या बालन मास पूरी वाला सी चालू करने वो रेगुलर था उसका but she had applied that knowledge there and हम कहाँ पे पहुँच गए? How data is there, knowledge is there, things are happening in front of you, but how you take it ahead, that makes lot of difference, lot of difference. वो अपना ये भी देखा आपने 3 Idiots, फुंसुक बाबू, लेह लद्दाख, simple things, utilization, हम भी तो साइकिल चलाते हैं, but हम कभी इसका यूज अलग तरीके से करते हैं क्या? Please come out of this shell which you all have created around on your own. Because your IQ is more important than your research में हो जाता है. I did my research, my PhD for 9 years. कभी-कभी ऐसे ये आ जाते हैं दौर में कि भाई लगता है कि क्यों कर रहे हैं, कुछ हाथ में नहीं आ रहा है. But that last door, to open that last door, you should have that patience to open that last door, because that last door will be the answer. So what Robin Sharma said, it's not my saying. Robin Sharma says that सर से बात करें.
Thank you so much, respected ma'am, for that insightful and engaging session. It was truly a great way to kickstart our conference. I'm sure we all took away something valuable from the presentations, the new knowledge, perspectives, and even inspiration to apply in our own field of studies. And we definitely now believe that ICANN is more important than our IQ. Thank you once again, respected ma'am. May I now request Principal Professor Dr. Trupti Sharma ma'am to felicitate Professor Dr. Nirmal Milan Kasegar ma'am. Thank you so much, respected sir and respected ma'am. We'll be next having our online sessions. And to introduce our next guest speaker, may I now request Ms. Vidya Kakkad ma'am to give the introductory speech.
आजकल ये चल गया है। सब जगह हो गया। दिल्ली में भी मैं साहित्य अकादमी के शेयर कर रहा था। एक उन्होंने कहा कि मैं ऑनलाइन हम अभी सिलेक्शन कमेटी में बैठे हैं। सिलेक्शन कमेटी में कैंडिडेट कहता है कि मैं ऑनलाइन आऊँगा, नहीं आ पा रहा। मैंने कहा आज तो क्या कर सकते हैं, टाइम ही नहीं है। तो दूसरे सेंट्रल यूनिवर्सिटी में बैठा था, बोले उन्होंने कहा कि क्या कर सकते हैं, दे दो कर दो, अच्छा लगेगा। तो इंटरव्यू तक में सीरियसली नहीं लिया उसने। इंटरव्यू बोलो ना क्या करना इसे नहीं, उसका सिलेक्शन नहीं किया जा सकता था। क्वेश्चन क्या होता है कि रिसर्च तो कर स्पेसिफिक पे, लेकिन टीचर बनने की क्वालिटी है। रिसर्च टीचर के साथ तालमेल हो जाए अच्छी बात है। इतनी अच्छे रिसर्चर, ये भी कहा गया, वाइस चांसलर ये भी कहते हैं कि वो अच्छा एकेडमिशियन हो सकता है, लेकिन एडमिनिस्ट्रेटर तो जरूरी नहीं हो। चार नहीं तालमेल में जिसका अच्छा तालमेल हो सकता है, लेकिन वो अच्छा एकेडमिक एक्सीलेंस नहीं है। दोनों को तालमेल चाहिए। टीचर के लिए चीज़ इम्प्रूव करना है, टीचर अच्छा भी बने, टीचर नॉलेज हो, रिसर्चर हो सकते, लेकिन जैसे लेकिन नाइबा
Before we get into the sessions, may I request Principal Professor Dr. Tripti Sharma ma'am to felicitate our respected Rajan sir. Let's give a big round of applause. Thank you, respected sir. Thank you, ma'am.
बहुत अच्छा बना। जाना चाह रहा हूँ
Welcome, Dr. Professor Supriya Sri Hari Siddhartha. Good morning, distinguished guests, esteemed colleagues, and respected attendees. It is with a great honor and privilege that I, Vidya Kakar, Assistant Professor, stand before you today to introduce visionary leader, exceptional academician, and researcher in pharmaceutical sciences, Dr. Professor Supriya Sri Hari Siddhartha. Dr. Supriya earned her B.Pharm in 1988, M.Pharm in 1990, and PhD in Pharmaceutics in 1994 from the Bombay College of Pharmacy, University of Mumbai. Her academic excellence laid the foundation for the distinguished career in pharmaceutical research and education. With outstanding 31-year career in academia and research, Dr. Supriya lead as a principal of Vivekananda Education Society, College of Pharmacy, Mumbai since 2009. In this, she trained 29 batches of B.Pharm students with exceptional success rate, launched M.Pharm and PhD program, and mentor over 41 M.Pharm and PhD students with several ongoing research projects under her with guidance. Her expertise extends beyond the Mumbai, with research guide approval from Bharati Vidyapit, Names University and Paher University, Udaipur. Dr. Supriya contributes pharmaceutical research are nothing short of extraordinary. She has successfully secured over 1.26 crore in research grant along with 59,000 euros and 8,000 US dollar from international collaboration that highlight her global impact in drug delivery innovation. She has published 64 international research papers and present 56 papers at prestigious conferences and has 5 granted patents that indicate her dedication in pharmaceutical advancement. Her contribution extended beyond the academia into the industry collaboration, leadership role, and prestigious recognition. She serving as a scientific advisor for innovative drug formulation, referring M.Pharm and PhD students thesis for top the university, being a reviewer of for international journal like Journal of Pharmaceutical Innovation, chairing and participating in syllabus committee at the University of Mumbai, judging in live competitions such as Avishkar, Inter-University Research Convention, being invited for round table session with Dr. Margaret Hamburg, Commissioner of US FDA, a recognition of her status in global pharmaceutical science. From her groundbreaking research in targeted drug formulation to her leadership academia, she is going to give the glimpse us into the topic of revolutionizing personalized medicine. I am sure ma'am is here to take us in insightful journey into the future of pharmaceuticals. All the students and the dignitaries, please join me in giving a warm welcome to Dr. Supriya Sriharisida. Welcome, ma'am. Over to you, ma'am. Ma'am, please check the mic. Ma'am, you are not audible. Please check the mic.
Thanks a lot. In particular, Dr. Sharma is present Institute of Pharmaceutical Sciences,
Sorry to interrupt, ma'am. Please take a pause for a moment.
Hello. Hello, ma'am. Just say hello. Okay, okay, okay.
Ma'am, sorry to interrupt. Ma'am, we'll just clear the issue. We just have a little technical problem. Ma'am, sorry to interrupt. Ma'am, am I audible? We are just clearing our technical issues. We'll get back to you. We'll just take a few minutes, ma'am.
Hello. Ma'am, am I audible?
Can you hear me now?
Yes, ma'am, you're audible now. Ma'am, could you please speak?
Am I audible? Can you proceed?
Yes.
Hello. Can I proceed with the talk?
Ma'am, just a second, ma'am.
Can I proceed?
Yes, you may please.
Hello.
Yes,
Am I
ma'am.
audible?
Yes, ma'am, you may proceed.
Hello. Voice is not listening. Let's see. So mute and I'll be able to talk. And just so that screen is visible. Is screen visible? Okay, so in the I'm just starting with my topic on personalized medicine, leveraging AI-driven 3D printing for tailored drug delivery system. In between, if I am not audible, please let me know. So I am going to cover 3D printing and various techniques, artificial intelligence in formulation development, personalized medicine and need and importance of it, role of 3D printing in personalized medicine, role of AI in personalized medicine and integration of AI and 3D printing in personalized medicine. So 3D printing technology is emerging rapidly. It is also called as additive manufacturing.
It is a process in which a material is added layer by layer to obtain the final object. So you require a computer aid. So computer aided design is that model which is constructed using software. For slicing also there is a software, and then there is a transfer of this programming operator and the 3D product is printed. So each 3D printing technology uses different materials, deposition techniques, layering manufacturing mechanisms, and final product characteristics. When we talk about the history of 3D printing, from engineering to medicine, this 3D printing was not new in the engineering, and slowly it got translated for its application in the pharma. So the history starts from 1980 till date. So in 1980, commercialization of 3D printing happened, but not for pharma results. In 1984, SLA, that is it is stereolithography that was invented. In 1990, utilization of inkjet powder bed was tried. In 1992, fused deposition modeling, that is FDM, was patented and it was developed by Scott company. Then in 2003, demonstration of inkjet-based 3D printing was done for viable cells. In 2005, successful creation of scaffolds mimicking bone structure was achieved. So it was its application for tissue re-engineering. Then in 2009, a functional vascular structure were engineered using bioprinting. So for these tissue engineering, there was a piston technology that was used, that is a pressure assisted micro needle technology that was used. Then in 2014, validated 3D printing for personalized medicines and innovative drug was tried. In 2015, first 3D printed drug, Sprita, was approved by FDA. In 2016, 3D printing for on-demand personalized dosage forms was implemented. 2017, successful development of 3D printed FSR tablets were developed. In 2019, oral tablets, that is caffeine tablet, were manufactured, highlighting the potential of MPL, fused deposition modeling, in personalized treatment. Then in 2022, self-nano-emulsified tablets, dapagliflozin, was made and was developed for, that is, this was a poorly soluble drug. So this particular technology was extended for improving the solubility of poorly soluble drug, and also it was extended to nano formulation, thus enhancing the bioavailability of the drug. So what is this 3D printing for pharmaceuticals? So there are various techniques that are very commonly used for 3D printing. The first one is fused deposition modeling. What is it? It involves extrusion of melted polymer filaments that contain the drug. So it is fused deposition modeling. There is a need of pre-formed filaments containing the drug, and then layer by layer they are deposited to form a tablet. So it is suitable for tablets with different shapes and layers. Then stereo.
So this is a laser-based writing technique. So it uses laser to cure
Liquid resin into solid structure. So it is used for highly detailed and precise dosage forms. Then this is, as you see here, it is utilizing resin, and hence it will have its own limitations. But this is based on the UV radiations that are used for photopolymerization of resin and converting the melted liquefied resin into the solid bed. Then there is a selective laser sintering technique which uses a laser to fuse powdered materials. Okay, so here we are using it for making porous structure that can control the drug units. And there is something called as inkjet printing. This was first tried, and this is a very easy to understand. So it deposits bits of drug containing solution onto a substrate. So your drug can be present in the liquid or drug can be present in the powder bed, and the liquid solution containing the binder will be deposited on that powder bed, thus allowing the second layer of powder bed to stick over the first bed, and that is how layer by layer the deposition of powder bed takes place till you get the required thickness, followed by cutting into required geometry. So this allows precise control over the distribution of drug within the population. So these are the two major classes of 3D printing technologies. We call it as nozzle-based deposition, and that is one class. In nozzle-based deposition, we have FDM, that is fused deposition modeling. We have DPE, that is dry powder, that is direct powder extrusion, direct powder extrusion. Then we have SSD, that is semi-solid extrusion. And under nozzle-based deposition, there is one more type which is called as PAM, pressure assisted micro syringe. Okay, so these are all nozzle-based depositions. So what are the factors to be considered while designing the formulation? So always remember, when we say ink, it is a pharmaceutical ink. What do you mean by pharmaceutical ink? It comprises of some polymers, some binding solutions, solvent, a blend which is malleable, which can be extruded. That is the pharmaceutical ink. So in fused deposition modeling, one of the limitations is that you require a pre-formed filament. So there is a technology and lot of factors required to form that filament. And the parameters which decide the success of this technology would be flexibility of this filament, hardness of this filament, density, temperature of this filament. So generally, we need to know the glass transition temperature of the polymer, and this particular process takes place at around 30 degrees temperature higher than the glass transition temperature, but it should be the temperature less than the melting point of the polymer. So at this temperature, this polymer is not melted, but it is malleable, so that it can be extruded and it can acquire various geometrical sizes. Generally, it gets extruded in the form of filament, and we coil it, and while coiling, we deposit layer by layer. Then DPE, that is dry powder extrusion or semi-solid extrusion. So this particular method has the advantage over FDM in the sense it doesn't require a filament. Here you have a direct powder mixture which is blended with the polymer and solvent, and then that powder blend is heated at a temperature 30 degrees above the glass transition temperature of the polymer, and then this malleable mass then extruded to get the cylindrical shape of the extruded which gets deposited. So here also heat is involved. In both the methods, the temperature is high. So these methods are not suitable for thermolabile drugs, and the factors that you need to consider are density, temperature of extrusion, right? But if you want to handle a thermolabile drug, you have a rescue, and that is this method, pressure assisted micro syringe method. So here there are pre-filled syringes which contain gel-like polymeric solution, and directly with the help of a plunger or pneumatic pump, that is with the help of compressed air, the mass gets extruded, and the parameters that are required to be considered here for the gel-like material is viscosity, rheological properties, and homogeneity. So this particular technique is quite popular for tissue re-engineering, or preparing the scaffold of cells, for preparing the for growth of cell to make it a organ on chip like device or such thing. This particular technique is quite popular. You can use it on thermolabile because high temperature is not important. Then we go to this side. These are laser-based scientific techniques. Laser UV of particular wavelength is used as a laser technique. And here we have a resin as a raw material. So this resin is dispersed with the drug, and this mask contains the photo initiator. So when the UV is activated on this mask, the photo initiator gets activated. It generates the free radical, and the free radical mechanism is involved in polymerization of that resin, and the liquid mass starts getting solidified. Because the drug is dispersed there, the drug also gets entrapped into that solidified mass. So here the parameters that govern the success of this technique are the power of the laser, the wavelength of the UV light, and time of exposure. So you will be having a software, G-code software, to ensure that irradiation is happening at particular coordinates so that you can decide what extent you want solidification and whether you can have the geometry based on different sides that are getting irradiated. Then we have a sintered laser, sintering technique, SLS. This also involves UV light and it requires powder mixture. And here the parameters are same, that is power of laser, UV light, and dynamic exposure. There is one technology which is called zip dose technology, which is a very famous popular technology called orodispersible tablet. This technology is based on 3D printing. So it employs 3D printing to layer a watery fluid over powdered medication, which rapidly dissolves upon contact with the liquid. So it arranges and binds the powder layers with a binding fluid, resulting in a highly porous dosage form that disintegrates upon contact with small quantity of liquid. So first step is that a powder blend is deposited and single inlet on the platform. This is a conveyor belt platform where the powder blend of drug and polymer. So a binding fluid is then drawn from the nozzle and it is deposited in the powder bed. As this liquid gets deposited, the surface becomes sticky, and then the next powder bed or layer will be deposited on it. Thus, these layers start getting deposited one after the other in the required thickness. So, the step 1 and 2 gets repeated several times to add more layers based on the dosage to form a film. And then a cutter which cuts the product into the required dimension. So this kind of highly porous dosing. I'm going to see this technology was used for first treatment product, and that was split up. Okay, so we will talk about it later. So this technology allows the production of high risk dose medication of 2000 mg, which is a very high dose for an orodispersible tablet. So orodispersible tablet has one limitation, that the drug dose has to be small, about 25 mg or less. Whereas this technology is allowing incorporation of high dose medication of 2000 mg. That's a great application of this technology. Scaling up is possible by using multiple nozzles that deposit the binding solution on the powder bed and which is transported on the conveyor belt. Tablets are gradually built layer by layer until they are completed. You can modify the technology by integrating it with the blister also, so that this powder bed layering is happening directly into the blister, followed by lining of the blister as a last step. So the product is formed, the tablet is formed within the blister. And that is a orodispersible tablet. So it is quite fragile. Its hardness is very low. So it is a peel-off type of blister and very softly you have to take the product out and put it in the mouth. Within no time it will get disintegrated, and thus this is going to be a orodispersible type of technology. So as I told you, split up was the first product approved by FDA developed this technology. This is by abrasion on speakers, and the drug is levetiracetam, that is an anti-epileptic drug, and this is giving you a very short integration time of less than 1 minute. This product is available in various strengths. So this is the pioneering effort by the company of Asia Pharmaceutical first to use India food product. There is one product called T90. This is a Chinese pharmaceutical company which has launched it, and this is also done with 3D printing technology. The company's name is Diastate. So it has received investigational new drug approval from the US FDA for its first 3D printing drug product. So when we say it has got IND, it is still under clinical trials, it is not yet launched. So it is prepared using technical depository. It has a specific shape with a precise internal geometrical structure that facilitates the precise control of the drug's release to achieve the desired uptake by the patient. So what we can see here is that not just that this drug of required dose you are incorporated, but also you are able to precisely control the drug release rate, right? And it is taken, what kind of precise control? It is taken at the bedtime and the drug will be released in a delayed manner. So this is a delayed release formulation. You may call it as pulsatile release formulation, but it is single pulse. So we have called it as, it is called as delayed release formulation. So the formulation will be taken at the bedtime, but the drug release will happen at early hours in the morning, where symptoms of pain and joint stiffness are very high because of circadian rhythm. So this product is under now clinical trials, and very soon it will come to market. So there are many formulations which are developed using 3D technology and reported in the literature, and they are covered anti-hypertensive drugs, anti-asthma drugs, then Sildenafil, which is again for pulmonary hypertension, propranolol anti-hypertensive, proton pump inhibitor, omeprazole, methotrexate, anti-cancer drug. So all these drugs have been tried using various 3D printed technologies like fused deposition, selective laser sintering, semi-solid extrusion, and so on. Right? And if you see the type of formulation, we have delayed release product, fast release product, cold shell printed, sustained release profile. Then we have floating type of product, and chewable type of product, and nanoparticle product. Okay, so this was about 3D printing. Now something about artificial intelligence. So artificial intelligence is playing its big role in drug discovery, prediction of pharmacokinetics of API. You all might have heard about the software Cascode Plus. If you have not read about it, just Google out and read about Cascode Plus. You will come to know something about it, the preliminary information which will let you know that based on the data, in vitro data, resolution testing, the software will be able to predict the pharmacokinetics of the drug in terms of its Cmax, Tmax, the rate of absorption, area under the curve, clearance. So everything will be predicted, which usually you require preclinical and clinical trials. But the software will be able to give you this data in vivo, so prediction of pharmacokinetics of API, prediction of toxicity of drug candidates. Again, for toxicity studies, you require acute toxicity of animals, but these models, prediction of pharmacokinetics of API, prediction of toxicity of drugs and candidates, at this moment, we will not say that we can wait for preclinical studies and clinical studies. But what we can say is that the success rate for these in vivo studies will be very high when you are able to predict the pharmacokinetic parameters using these softwares, AI-driven software. So very less chances that your preclinical studies, clinical trials will fail, and you will come to know well in advance what kind of parameters you are going to get, and the predicted and the actual parameters. There is a huge data to show that there is actually a good correlation, very high correlation. What you are predicting and what you got actually, that is almost similar. So that's the wonder of such type of AI-driven softwares and AI-driven technology. Formulation development, this is also possible because it is the software, the AI-driven software which has an algorithm which is going to predict what kind of suitability of the API is required, what kind of drug excipient compatibility is expected, what excipients can be screened based on that compatibility. Then it also will optimize various parameters, that is levels of polymers, levels of excipient source, level of drug polymer ratio that is anticipated in the sustained release product. Also, it will be able to give you some prediction of the processing parameters and how can you optimize it in terms of the rate of extrusion, temperature, while drying, while mixing, while blending, the other process parameters that are important in the manufacturing. Everything will be predicted, and if we have predicted everything, then the process of development becomes so fast, it's going to save your time, and it is also going to have economic implications because of saving of time, no trial and error method, no testing of raw materials, and hence a great success. Drug repurposing is possible using computer-aided drug designing, so you have to know a particular drug having particular kind of pharmacological action can have affinity for some other receptors also. For example, sildenafil. It was used for erectile dysfunction, but it was repurposed. It was used for as an anti-arthritic drug, but it was repurposed for cancer also, or something like anti-diabetic drug. So there are many such examples. The repurposing is possible, and this is because of drug discovery tools that we have, the computational program modeling, molecular modeling that we have, which is AI-driven. Then personalized medicine, that is precision medicine, that is also possible using AI tools. So AI, that is artificial intelligence and ML machines. These are all useful tools for predicting formulation performances. So AI algorithms which are available are artificial neural networks, support vector machines, and random forests. And they can predict solidity and salinity, bioavailability, dissolution rates, mechanical properties. Then data analysis. There also, so AI algorithms require a lot of data. Okay, so data analysis is a very important step when designing the algorithm. So you have a huge data, vast data, which helps you to identify certain patterns and relationships that might be missed by human researchers. So it is the machine which is able to tell you that this is going to be a trend because the data is showing this kind of trend, this kind of future action or future path of that particular process. So researchers may miss it, but machine will not miss because it is computerized. It is using a statistical tool, and that can lead to the better formulation design. So formulation optimization I have already spoken about. So you all know the pharmaceutical process validation. It requires formulation optimization. You all have learned about the really quality by design, where the important tool that is used is design of experiment, and the design of experiment is based on various designs. So these are all AI-driven designs, software-driven designs. So one is Bayesian optimization, which can find the optimal combination of formulation parameters. For example, excipient types, concentration, processing condition involves time of exposure, temperature of processing, the speed of rotation of that particular process, etc. So this helps to achieve performance characteristic. So all these are independent variables which are under the control of the formulator, and this is that optimization design which helps in optimizing these independent variables. There is a generative model which is very popular nowadays. So these models can be used to generate new formulation and design with specific properties, accelerating the development process. Reinforcement reinforcement learning. This is the approach which can be used to train AI agents to optimize formulation processes in real time, leading to more efficient and robust formulation. So it can be used to train AI agents to optimize the formulation. That is the important learning. That's a learning tool for AI agents. Now, specific AI tools and platforms are like summarized here. Formulation AI, a web-based platform for drug formulation design that uses AI to predict and optimize formulation. BioPR formulation design is a cloud-native solution for managing recipes, ingredients, domain-specific calculations to optimize formulation. Addition and AI-based tools to identify potential pharmaceutical ingredients. So you can see for every type of step in formulation development, there is some software based on AI available for you. So this is going to completely eliminate any trial and error method, and there is going to be a very scientific and rationalized approach for designing of the dosage form. There is deep getting or deep thoughts or deep development with AI or organic AI tools available. These are examples of tools using drug discovery and development that can also be applied to formulation development. So all those who are having research interest in chemistry and drug discovery, these tools will come across very often, and that will help in computer-aided drug designing, which will have application for formulation development also. Because, for instance, going for test masking, then you have the molecular modeling, whereby you are creating the in silico test receptors, and you are checking whether there is any molecule available which can block the test receptor, and thus it can lead to test masking for a while till the bitter drug is consumed. Or you can have in silico modeling for drug ligand conjugation. So this ligand can have a specific receptors in the body, and the drug can be targeted to a specific receptor for which you require a ligand, and such conjugates can easily identify in silico using any one of these softwares. So we have a lot of applications even for formulation development. The world is of collaboration. So you can see these are all major pharma industries: Pfizer, Novartis, Roche, Bayer, Sanofi. They are all multinational players, very big companies, and you can see they all have joined hands with AI, ML based engineering companies, so that they collaborate to have combinations of quantum mechanics and machine learning, crystal structure prediction, and screening using computer models, research in immune oncology as an approach to cancer treatment that uses body's immune system to the fighting with the cancer. So what we see is that these are AI-based organizations. They are doing research in AI, and these are pharmaceutical companies, and there is a collaboration which is leading to a product which is designed using AI technology. So formulation development, the process development, continuous manufacturing, quality control of product, quality assurance, documentation, everything is possible by making use of AI ML, for which they require partnering, and they have partnered with these companies which are expertise in AI. Then talking about integration of AI technology in printing, we have seen AI, we have also seen 3D printing. How do we integrate it together so that we get a product which is the precision based, which is having more beats, it is preventive, it is predictive, it is participatory, and it is also giving you a proper procedure of those, right? So design optimization is possible with generative design that we saw. So AI algorithms will generate and optimize design based on unique parameters and constraints. So AI will be used for optimizing the formulation, for developing the formulation, and then process monitoring, for which also AI will be used. So product will be actually produced using 3D printing technology, but But the formula is required to be designed before you put it into 3D printer. The formulation should be designed. It should have polymers, it should have excipients, it should have drug, the proper ratio, it should have some binding solutions, dilutes. That formula will be designed using AI algorithms. Not just that, the 3D printing technology requires a close monitoring, real-time monitoring, AI-powered sensors are required, cameras are there, which are displayed on the 3D printing process for real-time photograph. And also the sensors which are telling you that a proper thickness is getting generated, proper stickiness, viscosity is maintained. And if you get a signal alarm, you need to stop there because the sensors are telling you that something wrong has happened. The process is not going as per the expectation, there is some change in the parameter. So these sensors are there which will help in real-time monitoring, and thus it is an IPQC, in-process quality control, that you are able to achieve, and that will help in ease failure rate. You have a preventive maintenance strategy, and that will help locate deviations, mistake, and enable instant adjustments. So predictive maintenance, I spoke about preventive maintenance. So predictive maintenance, examine data from printers to predict maintenance desires based on the utilization style and performance. So this is how AI has helped us in the printing process. Quality assurance is also possible. I spoke about IPQC. It is a part of quality assurance. Right? So computer region, computer, the sensor, which is with the product, which is continuously monitoring the product. So AI-driven computer vision inspects the printed objects for excellent control. That is how we have integrated AI with the 3D printing technology. So, huge benefits we are able to achieve. We have personalized medicine, complex drug delivery systems are possible. How complex? I am showing you a couple of case studies. Rapid drug development, you have totally shrunk the product pipeline time period because of AI. Everything is predicted beforehand, and the failure possibility is reduced. So definitely the product life cycle for development is reduced, and in less time we will produce a more precise product. Then manufacturing is possible on demand, that is a precision medicine. Medicine customized as per the genotype, phenotype of a patient. That is manufacturing on demand, cost effectiveness. So see, when we talk about cost, it is definitely cost. But cost efficiency in what sense? That the person is getting cured, the failure rate is less, the effectiveness is improved, and because of this procedure, based on its genetic makeup and his blood work and urine reports and his DNA, the so it is not the one formula which is applicable to all, and for some it is effective, for some it is not. It is about a guaranteed formula which is developed for a patient. So it becomes cost effective, right? It has to be in cure, and that is how the quality of life is improved in a shorter period of time. But if you consider a single product, then it is going to be very costly if we are using 3D printing technology, which is for precision medicine, not for startup. Startup was something which was a oral disbursement tablet, which was not precision medicine, and it was made using 3D printing technology. But if you are using 3D printing technology for precision medicine, that is customized medicine based on patient need, it is going to be expensive. Optimization and patient population will be always very high because it is optimized for a particular patient based on his lifestyle, his age, his other comorbidities. So every patient population. So that is what I was talking about. Suppose we are talking about cancer patients having colon cancer, and this is the therapy, that is one treatment which fits on. Then few of the patients will show effect, few will not show effect, and few may show adverse effect. But what is the futuristic medicine? It is a more personalized diagnostic medicine. So it is based on the genetic setup, the biomarkers, the lifestyle of that patient. So it is bound to give you personalized medicine will be giving you effect. The therapy will be giving good effect. So there is a need for personalized medicine. It is based on the genetic variability. Patient will be responding. Patients have different genetic makeup, but your medicine will be varying based on the genetic makeup. There is going to be improved drug efficacy, tailored treatment which ensures better therapeutic outcome. So there are reduced side effects because you are minimizing the adverse reaction by considering patient-specific drug metabolism. Why sometimes side effects happen? Because drug doesn't get metabolized, drug doesn't get eliminated in some patients. Maybe because their liver is compromised, there is less level of metabolizing enzyme, the kidneys are compromised, the renal glomerular filtration rate is reduced, and so there is a retention of drug which is giving side effects. So everything can be avoided. Customize the medicine based on patients, phenotype and genotype, and entire lifestyle and overall anatomical physiological conditions. Chronic disease management is also possible, like cancer, diabetes, cardiovascular diseases. So, AI-driven data analysis will help you in developing the algorithm and 3D printing to get your precise information for each patient. So, it is going to help you in enhancing treatment, new style and error approach, and early detection of risk enables proactive treatment. So it is like we are able to identify to what is that predisposition of the patient to a particular disease. This patient is predisposed to a particular disease, and this you are able to identify before he encounters that disease based on the biomarker and genotype, the genetic structures of the DNA. And then, before he encounters the problem, the preventive measures we are able to take, and that early detection will reduce the risk, and thus it is a proactive treatment. So AI is primarily its role is to process vast amounts of diverse data, including genomic sequences, medical images, electronic health records like MRI, CT, and patient-reported outcomes. The genomic data analysis to identify genetic mutations, variations that may predispose individuals to certain diseases, and predictive analytics, which will forecast the likelihood of disease progression or relapse in patients with chronic conditions. So it is really a great tool for patient because this analytics is helping to find out what disease you are exposed, and this analysis, predictive analytics, will help prevent the progression or relapse of that disease in case of chronic conditions. So, first to introduce the anti-personalized medicine solution is by Bush, and that was for cancer treatment. The subsidiary of that company, Foundation which uses AI in analyzing tumor samples to identify mutations related with particular treatment is received. We have seen how it has impacted the mutation in the tumor. And these are various algorithms which are the deep bio. So this is used for prostate cancer detection. The DIA-D, which is the algorithm developed for breast cancer biomarker quantification. StratiPath algorithm for risk profiling for invasive breast cancer. And UniGate algorithm for screening and training for prostate cancer. Analyzing link metastasis and screening for uterine cancer. So 3D printing is a personalized medicine design steps that design the 3D structure using a computer-aided design software or obtain a geometry using 3D scanner to create the STL file. So you require the molecular modeling software or a 3D scanner, 3-liner, particularly develops a particular geometry. Molecular modeling develops a particular design, and then there is a STL file that is developed. This HDL file is then sliced into different layers, and these layers are superimposed on top of the other during the actual printing that happens to develop the joint. So for slicing also, there are softwares available so that with what precision the slicing should take place outside, and also this helps in a better resolution. So there is a G-code software which helps in generating that is generated that can be sent to the printer, dictating the exact coordinates where the mouse will be deposited. So this G-code also has a very big role to play because this combination adds the cylindrical filament that is getting deposited. You here also, or maybe any other technique where laser granulation is taking place, you need to know exactly at what coordinates the intensity of laser should be high or low. Or you also need to know if you are through nozzle putting the drug solution at what coordinates the quantity should vary. So the G coding will decide the precision of this kind of printing at various coordinates, x coordinate, y, z. So this is a 3D model, right? So various coordinates are there. So where to deposit what amount of ink that we decided by G code. And when I say ink, I mean pharmaceutical ink. It's nothing but the blend of its ink. There is a concept of polyfill. So polyfill was launched by Canada Pharmaceutical in 2009. That was not based on but that was a concept which was very innovative. It was a capsule which was a single unit dosage form containing the medication for number of disorders that the patient is undergoing. There was a drug for treating anti-hyperlipidemia, drug for treating hypertension, and the drug for diuretic, causing diuresis. So you are taking care of heart, blood pressure, and hypertension level by giving only one dosage unit to the patient. This is the useful for geriatric patient, and that will not only improve his quality of life, because not only medicines he has to consume, only one formulation he has to consume, but also it will improve the patient's compliance because missing component medicine will be only one product we have to consume. So that was a polycare approach, and for which Canada Pharmaceuticals were received a lot of recognition. They were actually helping to improve the quality of life. Now, taking a kind of trigger or a motive from kind of lead from this kind of concept, the 3D printing polyfill also are now developed, and there is a research available on this. So this 3D printed polyfill will be tunable for drug release based on modified drug loading and excipient composition in the separate parts of the formulation. So the geometry of the polyfill was designed using the 3D drawing package polycap. So if you see this diagram, see the upper layer has two drugs, ACE, salicylic acid and hydrocortisone. Sorry, hydrochlorothiazide, that is diuretic. So this is acetylsalicylic acid. This is the blood thinner, acts like an anticoagulant, and hydrochlorothiazide, which is acting like a diuretic. This is present in the top layer, and it is an immediate release compartment. And if you read the paper on this, you come to know it has a super disintegrant sodium starch glycolate, and the other layer is comprising of three different compartments. In one compartment there is atenolol, and then in another is pravastatin, and third ramipril. So atenolol and ramipril, this is the S antagonist. Atenolol, this is the beta-1 that's the anti-hyperlipidemic drug. So what they have done, they have used cellulose acetate filament to make the segments in this layer. 3 segments are made in this layer, and in each segment the blend of the drug with HPMC and some plasticizers like polyethylene like manufactured. That was added in 3 different segments. The segments were created using the semi-permeable but hydrophobic polymer cellulose acetate. So what happened, you want a different drug release profiles for 3 different drugs, and this kind of product was done using fused deposition modeling. So it was quite hard enough to give you sustained release action, and this entire part was meant for control release, and the upper layer was meant for sustained release. They also created the dots here at the top, which gives for geriatric formulations of E of the so by touch also you can identify the right formulation is there here, and that gives identity to that product. So this is such a great technology. This was developed by this company, and this 3D drawing package was the CAD, and this kind of drug is profile we got is polypill. So you can see in 30 minutes you got the immediate release of these 2 drugs, acetylsalicylic acid and hydrochloric acid, diuretic and blood thinner. And this sustained release profiles you can see that you have got for atenolol, pravastatin and lamivudine, which are antihypertensive and antihypertensive drugs. So this is how AI-driven design algorithms analyze vast data sets to predict optimal drug formulation, considering factors like solubility, stability, and release kinetics. This accelerates the drug development process and improves the efficacy of the final product. It is 3D printing enables besides layering of our materials to create. So this was AI tool, which is helping in all predictions, formulation, development prediction, and process development prediction. Then 3D printing, that's actually manufacturing technique. So it enables precise layering of material to create complex dosage forms with customized shapes, sizes, and drug release profiles. This level of control is unprecedented in traditional pharmaceutical manufacturing. And personalized medicine, by combining AI and 3D printing, the personalized medicine becomes a reality. Tailored formulation can be created to match individual patient's needs, optimizing the therapeutic outcomes and minimizing the side effects. So for pediatric formulations also, we have challenges of taste masking, then the right geometry, difficulty in swallowing. So all these challenges are addressed by AI integrated solution, so that you can overcome taste masking problems, you can have good chewable type design or dispersible type of formulation, and thus you can improve the quality of life of a patient, pediatric patient, with better compliance to the treatment and more accurate treatment, thus relieving the stress of the caregiver. Control release implants are available for chronic diseases, which are helping to maintain the drug level in the body, which is very critical for chronic diseases like maybe chronic respiratory problems, chronic diabetes, something like chronic hormonal imbalance problems. So these are the kind of implants which can slowly release the drug and thus prevent the fluctuation of drug in the blood, and also it is the implant will have sustained action, one implant per week, one implant per month. That is how you can reduce the frequency. So AI models predict optimal implant designs, material composition for sustained drug release over extended period. 3D printing enables the creation of implants with complex geometries and manipulated structures. The outcome is that there is improved therapeutic efficacy, reduced dosing frequency, and enhanced patient quality of life through consistent drug delivery. So transfer patches for which also this the AI printing is applicable. So for transfer patches, wardrobe condition is the biggest challenge.
अगर ऑफ करना है तो मेरे को सब बंद करना पड़ेगा। मैम को बोलिए ना अपर्णा मैम वो कर सकते नहीं।
So we have this
अब
technology
देख
which
ये
can
हिल रहा है
create the
तो
micrometers. There are some good
अब
risks
देखिए
that
ना
are created
ये
using this technology. So AI
मेरी मैम
algorithms
से बात करके
identify permeation enhancers.
इसको
So
कैमरा पे लगा,
population
मैं मैम से
development
बात
is
करके आती
possible by identifying right permeation enhancer, optimize the patch microstructure to facilitate drug transport process, the skin barrier,